If you analyze single-cell RNA sequencing, ATAC sequencing, spatial transcriptomics, or multiomics, you already know where the bottleneck is. It isn't sequencing anymore, it's interpretation. In this video we'll go through the Python libraries that let you build AI directly into your analysis workflow, what each one is actually for, and how they fit together into a single reproducible pipeline. Most of us use AI like this. You hit an error, you copy the trace back into a browser, you paste the answer back, and you repeat. Every one of those handoffs loses context. The model never sees your data object, your metadata, or the decisions you made yesterday, and none of it is recorded anywhere. It works, but it doesn't scale, and it is not reproducible. It helps to see these libraries as 4 layers rather than a long list. At the bottom, model access. How do you call a language model at all? Above that, structure, forcing the output into a schema you can actually parse. Then retrieval and documents, giving the model your papers and your data. And at the top, agents, laying the model plan, call your functions, and check its own work. Each layer fixes one specific failure of the layer below it. The base layer is the provider SDK, the OpenAI Python library, and the equivalents from Anthropic and Google. All give you the same primitive: send a list of messages, get a response back. You'll use this for code generation, for interpreting statistical output, and for drafting methods text. But notice the risk. If you hardcode one provider into 50 scripts, you are locked in, and that becomes a real problem the moment prices change or a better model appears. Light LLM solves exactly that. It wraps every major provider behind one single interface, so switching from one model to another is a one-string change. There are 2 practical wins for a lab. First, you can benchmark the same prompt across models on your own task, and actually measure which one annotates your clusters correctly, instead of guessing from a leaderboard. Second, you can route cheap high-volume calls to a small model, and keep the expensive one for the hard reasoning. Free text is the enemy of a pipeline. If you ask a model for marker genes, you might get a bulleted list, a paragraph, or a markdown table, and your parser breaks on the next run. Pydantic AI fixes this by making you declare the shape you want as a Pydantic model. The library validates the response against that schema, and if the model returns something that doesn't fit, it feeds the validation error back and retries automatically. You get a typed Python object every time. This matters more in genomics than almost anywhere else. Gene symbols are ambiguous and they change over time. A model will happily hand you a symbol that isn't in your reference, or an alias that was renamed 3 years ago. Now, a schema does not make the biology correct. What it does is guarantee you get a typed list, which you can immediately intersect against your var names, so a hallucinated gene fails loudly at that check, instead of quietly ending up in your results table. Next, retrieval. A model only knows what is in its context window right now. Retrieval augmented generation fixes that. You embed your documents into a vector index, find the chunks most similar to the question being asked, and put only those chunks into the prompt. Llama index gives you that entire pipeline: loaders, chunking, embeddings, the index itself, and a query engine. The important point is that the model never memorizes your data. It retrieves it at query time with a citation. In practice, pointed at everything your lab has ever written: papers, protocols, your own code base, meeting notes, previous analysis reports. Then a new student can ask why you chose a particular clustering resolution, and get an answer grounded in your own documents, with a citation back to the source file. In my experience, this is the highest value first project for most groups, because the payoff is immediate and nothing about your existing analysis has to change. But most of that knowledge is trapped inside PDFs. DocLink converts PDF, Word, PowerPoint, and HTML into clean structured markdown, and critically it is layout aware, so tables, headings, and reading order survive. That is what makes methods mining possible. You can parse 200 papers, pull out every methods section, and extract which normalization, which integration method, and which clustering resolution each one used. That's a review table you would otherwise build by hand over several weeks. LangChain is the plumbing layer. It standardizes how you connect a model to everything else: prompt templates, vector stores, SQL databases, APIs, and your own Python functions. The core idea is the tool. You take an ordinary function, you decorate it, and you hand it to the model as something it is allowed to call. And look carefully at that docstring, because that docstring is the only documentation the model gets. If it is vague, the model will use the tool wrongly. Tool calling is the concept that makes all of the agent frameworks work, and it is worth being precise about it. The model does not run your code. It reads your function descriptions, decides which one to call and with which arguments, and returns that request as structured data. Your process then decides whether to execute it and passes the result back. That separation is your safety boundary. You keep complete control over what actually runs on your machine. Small agents is the minimal version of that loop. Think, act, observe, repeat, until the task is finished. It is deliberately small. It's a few hundred lines of concepts rather than a framework you have to commit to. Use it for the repetitive jobs you keep doing, by hand. Validating sample sheets, checking that every FAST has a matching row in your metadata, or regenerating the same figure across 10 datasets. And always set stop conditions, maximum steps, maximum costs, and where relevant, human approval. 𝗀 AI takes a different angle, role-based teams. You define each agent with a role, a goal, and a set of tools. Then you give the crew an ordered list of tasks for a single cell project that might be the 𝗀•cy agent that reads your metrics and flags outlier samples, an annotation agent that maps marker genes to cell types, and a reporting agent that writes the summary. The output of one task becomes the context for the next. If your lab already has a written SOP, it maps onto this almost line for line. Autogen is built around conversation between agents, rather than a fixed sequence. One agent proposes an analysis, another critiques it, and they iterate until they converge or hit a limit. That is genuinely useful for hypothesis generation, and for code review of analysis scripts the model itself wrote. But this is also exactly where costs run away, because every turn resends the whole conversation. Always cap the number of turns before you leave it running. Pandas AI sits at a much simpler level. You wrap a data frame, you ask a question in plain English, and it writes and runs the pandas code for you. For quickly exploring 𝗀•cy metrics, sample metadata, or a differential expression table, it is genuinely fast, but treat it as exploration, never as a result. Always read the code it generated before you believe the number, because a wrong grouping will give you a confident answer that is completely wrong. Hiding face transformers is the gateway to pre-trained models, including the biological ones. Geniformer, 𝗪Sci𝗺𝗵𝗶, 𝗪Sci Foundation and 𝗪𝗶 are trained on tens of millions of cells, and they produce embeddings you can use for cell type annotation, for batch robust integration, or for in silico perturbation. They are not a drop-in replacement for your standard pipeline. Benchmark them against 𝗀𝗵𝗄 or 𝗬𝗏 on your own labeled data first, because performance varies a lot by tissue. So here is how the whole thing fits together. Backling and Llama Index build the knowledge layer from your papers and protocols. Light LLM handles model access and routing. Pydantic AI enforces a schema at every boundary, where data crosses between the model and your code. LangChain exposes your 𝗀𝗵𝗄 and 𝗪𝗏 functions as tools. And an agent framework runs the loop, 𝗀•cy, integration, annotation, differential expression, writing a log entry at every single step. That log is the thing that makes it science rather than a demo. Now some hard-won caution. First, pin your versions. These libraries change therapy as fast, and a notebook that ran last month may simply not run today. Second, log every prompt and every response alongside your seeds, or your analysis is not reproducible. Third, never send patient-identifiable data to an external API without checking your IRB and your data use agreement first. And fourth, validate every biological claim against your actual data. The model does not know your biology. If you are starting today, view it in this order. Week 1. Structured output with Pydantic AI on one small task. Week 2. A retrieval index over your own papers. Week 3. Wrap 3 functions you already trust as tools, and only then reach for a multi-agent framework, because agents amplify whatever is underneath them, including the mistakes. Build the workflow, not the script. Full tutorials and code are at multiomics.com. Thanks for watching.